Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Today we're diving into really one of the biggest questions in biology. What actually causes aging? Is it some kind of genetic program, like a timer counting down?
Or is it maybe just stuff breaking down, accumulated damage, wear and tear, increasing entropy?
Exactly. That's the core debate. Program versus damage. And before we get into how our sources tackle this, please, if you find this useful, take a second to like, subscribe, and share it with friends and family. It really helps support the Sand Science Experience.
Yeah, it does. So this debate, program versus damage, it's been around for, decades. What's new is how these super accurate aging clocks, particularly the DNA methylation ones, fit in.
Right, the Horvath clocks and others like them.
Precisely. And our source material digs into whether these clocks are evidence for a program or if they actually measure something else entirely.
Which is?
They argue the clocks reflect the rate at which random stochastic molecular damage accumulates. Think entropy, but measurable.
Okay, so the goal here is to give you the context to understand why, just because we can measure age very accurately, it doesn't mean aging itself is intended or programmed.
Exactly. Precision doesn't equal purpose.
Let's start with the clocks themselves. How do they work? The main ones use DNA methylation, right?
That's right. They look at specific spots on your DNA, these CpG sites, where a chemical tag, a methyl group, can be attached or removed.
And the pattern of these tags changes predictably with age.
Amazingly predictably, from before birth right up to people over 100 years old. They're so accurate. They can even pick up accelerated aging from things like smoking or certain diseases. They really are like biological speedometers.
Okay, and that accuracy, that predictability, that's what's reignited the whole programmed aging idea.
Exactly. It feels counterintuitive, doesn't it? If aging is just random damage accumulating, how can you measure it so precisely, almost like clockwork?
It does seem like a paradox.
And it's led to a couple of flavors of program theory. There's programmed aging in the really strict sense, the idea that we have genes specifically evolved to make us decline and die.
Which, evolutionarily, it's always seemed like a tough sell. Why evolve self-destruction?
It faces huge hurdles, yeah. So the more common idea now is maybe programmatic aging. This is a bit more subtle.
How so?
It suggests aging isn't actively triggered, but it's an unintended consequence. Like the developmental programs that build us just keep running past their use-by date and start causing problems. And overshoot.
Like ontogeny extended too far. And the link to the clocks.
Well, proponents point out that many of the CpG sites the clocks track are near important developmental genes. Specifically, genes regulated by the polycomb repressor complex 2, PRC2.
PRC2, those are involved in shutting down genes during development.
Exactly. They put the brakes on development. So the argument is maybe these regulators are also setting the pace of aging later on. They're controlling the clock in a sense.
Okay, that sounds pretty plausible on the surface. So why did the authors we're looking at disagree? Why push back towards damage and entropy?
They start with basic evolutionary theory. The idea that we don't need a program to age and die. All you need is for natural selection's power to fade after we reproduce.
The selection shadow.
Right. Once you've passed on your genes, there's much less evolutionary pressure to keep your body in perfect condition indefinitely. Maintenance becomes less critical from selection's point of view.
So we're not actively programmed to fail. We're just not strongly selected to succeed forever.
Precisely. And this leads to the idea that aging is kind of the default state. Think about it at the molecular level. DNA, proteins, they're complex molecules constantly being jostled by, thermal energy, Brownian motion.
They're inherently unstable.
Exactly. Staying alive, maintaining order, requires constant, energy-intensive repair and maintenance. If that maintenance isn't perfect, and it never can be perfectly efficient forever,
then decay is inevitable. It's the default.
The authors put it starkly. The default is thus an unstable and decaying genome. Staying stable is the hard part that life achieves.
Okay, but hang on. If it's just decay, why do different species have such vastly different lifespans? Why do humans live so much longer than mice? Doesn't that imply some genetic control, some programming of lifespan?
That's a key point. But the argument is that the genetic control isn't programming death. It's programming the quality of maintenance.
So longer-lived species aren't programmed to live longer. They're just better at fighting off the default decay.
Exactly. When you compare species, a major factor correlating with longevity seems to be the efficiency and robustness of DNA repair systems. Better maintenance equals longer life. It's about resisting entropy for longer.
Okay, that makes sense. What about individuality? Even within a species, people age so differently.
And that's another strong argument against a rigid program. Look at worms. C. elegans. You can have genetically identical worms, clones, living in the exact same lab conditions.
And they don't all die at the same time.
Not even close. You see up to a threefold difference in their lifespans. Same genes, same environment, wildly different outcomes. That screams random chance, you know?
Yeah.
Stochastic factors are huge.
A threefold difference.
It is.
Wow. And in humans, what about heritability of lifespan?
Well, estimates vary, but once you account for things like people tending to partner with others of similar health or background assortative mating, the purely genetic, inherited component of how long you live might be surprisingly small.
How small?
Some estimates put it below 10%, maybe a bit higher depending on how you calculate, but certainly not the majority.
So most of the variation in how we age comes down to what? Luck, environment.
It suggests a huge role for non-genetic factors.
Mhm.
Things like epigenetic changes, random DNA damage accumulating over time. Basically, that stochastic, messy reality.
Which brings us right back to the paradox.
Yeah.
If it's all so random and messy, how can the clocks be so precise? How do you measure randomness accurately?
Okay, this is the core argument of the paper. The clocks work because they capture the average rate of this increasing randomness, this rising entropy. Think of it this way. Individual molecular errors, like a methyl tag getting lost where it shouldn't, or added where it shouldn't, those are random events, stochastic.
Right.
But the overall rate at which these errors accumulate across millions of cells, averaged out, can be remarkably consistent and predictable over time for a given individual or species. It's like radioactive decay. You can't predict when one specific atom will decay, but you can predict the half-life of the whole sample very accurately.
So the clock isn't tracking individual random hits. It's tracking the average rate of accumulating disorder.
Precisely.
Hmm.
It's measuring the predictable increase in system-wide noise or entropy over time, driven by the imperfect fidelity of our maintenance systems.
Okay, is there a specific mechanism they propose for why this drift, this accumulation of errors, happens at such a measurable rate, especially at those developmental gene sites?
Yes. Remember those PRC2 sites, the developmental genes that get shut down? They are kept repressed by active maintenance of their methylation patterns. But maintenance isn't perfect. Errors happen. Crucially, the authors argue these methylation sites don't just drift aimlessly. They tend to drift towards a specific site-dependent equilibrium state, an attractor.
So even though the errors are random, the direction of the drift is somewhat predictable for each site, trending towards some baseline state.
Exactly. It's the failure of the active maintenance away from the tightly regulated developmental state towards a more default or equilibrium methylation level for that specific spot. And because this happens across many sites, the average trend becomes measurable.
So the clock is measuring the rate at which active regulation is failing and the system is relaxing towards a less organized state.
That's the idea. And there's evidence linking damage directly to clock acceleration. Take mouse models that have defects in DNA repair genes, like ARI-C1 or XPG mutants.
They age rapidly, right? Progeria models?
Yes, they accumulate DNA damage much faster than normal mice. And critically, their epigenetic clocks also tick much faster. They show an epigenetic age far older than their actual chronological age.
So more damage accumulation directly speeds up the clock reading.
It strongly suggests the clock is sensitive to and likely measuring the consequences of that stochastic damage accumulation.
Okay, so let's boil this down. The headline takeaway is,
The aging clock isn't a preset timer counting down our lifespan according to a program. It's more like a very accurate speedometer, measuring the rate at which our biological system is accumulating molecular disorder, losing regulatory precision, and succumbing to entropy over time.
that's a really different way of thinking about it, and it must have huge implications for how we try to intervene in aging.
Massive. If aging isn't a program we can just switch off, but rather the default consequence of imperfect maintenance.
Then interventions need to focus on improving the maintenance, boosting repair.
Exactly. It shifts the focus away from finding a single aging gene or pathway to turn off and towards strategies that reinstate regulatory precision and enhance the capacity to repair damage and maintain order.
Which fits with the progeria data defects in repair cause faster aging. So enhancing repair should slow it down.
That's the hope. And we see hints of this. For instance, work targeting things like the dream complex, which normally suppresses DNA repair genes in adult cells, activating that in worms or mice, seems to help counter stochastic damage and extend health span.
And what about the most dramatic example, cellular reprogramming? using Yamanaka factors.
That fits perfectly too. Reprogramming resets the epigenetic clock essentially to zero. But how? The authors argue it's not just wiping a slate clean. It's fundamentally restoring the cell's ability to repair DNA damage effectively and reestablishing the tightly regulated gene expression patterns of an early embryo.
So reprogramming works because it fixes the maintenance systems and tightens regulation, not because it reverses some timer program.
That's the interpretation consistent with this view. It provides a kind of blueprint. Rejuvenation likely requires restoring both genome integrity and epigenetic precision.
It sounds like the main message is we need to move past looking for simple programs and accept the complexity.
Yeah, the authors suggest our human tendency to see patterns and purpose might make us want to find an aging program, but to actually make progress against aging, we probably need to embrace the messy stochastic reality of biology and focus on fighting that inevitable drift towards disorder.
Okay, lots to think about there. Before we wrap up, you mentioned a provocative thought for our listeners.
Yes, so if life itself is fundamentally about maintaining order against the universal tendency towards entropy or disorder, and aging is essentially the gradual failure of that maintenance, the accumulation of disorder, what do you think is the single biggest evolutionary or maybe even physical hurdle that we face in trying to achieve truly long-term stable rejuvenation? Where's the fundamental bottleneck in reversing entropy locally?
That's a deep one. Definitely something to chew on. Thank you for walking us through that complex picture.
My pleasure.
And thank you for tuning into this deep dive. If this helped clarify the whole program versus damage debate for you, please do hit that subscribe button, like the video, and share it with someone who might find it interesting. Your support really helps the Sand Science experience keep going.